Now, we will learn about glucose catabolism. Oxidation of glucose to produce ATP is also known as cellular respiration, and it's involved four sets of reactions. The first one is glucose turned into pyruvic acid, where it is known as glycolysis. Then the pyruvic acid will turn into 2-acetyl coenzyme A. This process is called formations of acetyl coenzyme A. And the third reaction is called Krebs cycle. And then the fourth reaction in the cellular respiration is the electron transport chain. Now let's just dive in more depth on glycolysis. As we all know, glycolysis is a set of reactions in which one glucose molecule is oxidized and two molecules of pyruvic acid are produced. The reaction also produces two molecules of ATP and two energy-containing NADH. Because glycolysis does not require oxygen, it is a way to produce ATP anaerobically, which is without oxygen and is known as anaerobic cellular respiration. This chemical reaction split 6 carbon molecules of glucose into 3 carbon molecules of pyruvic acid. Even though glycolysis consumes 2 ATP molecules, it produces 4 ATP molecules for a net gain of 2 ATP molecules for each glucose molecule that is oxidized. There are 10 reactions of glycolysis. The first one is glucose is phosphorylated using phosphate group from an ATP molecule to form glucose 6-phosphate. Then the glucose 6-phosphate is converted to fructose 6-phosphate. A second ATP is used here to add a second phosphate group to fructose 6-phosphate to form fructose 1,6-biphosphate. And now let's focus here. The fructose splits into 3 carbon molecules. Here is 1 molecule of 3 carbon, and here is another 1 molecule of 3 carbon, so that the fructose 1,6-biphosphate split into 2 3-carbon molecules, the glyceraldehyde triphosphate and dihydroxyacetone phosphate, each having 1 phosphate group. Oxidation occurs as 2 molecules of NAD accept 2 pairs of electron and hydrogen ion from 2 molecules of glyceraldehyde triphosphate to form 2 molecules of NADH. Body cells use the 2 NADH produced in this type to generate ATP in the electron transport chain. A second phosphate group attached to the glyceraldehyde triphosphate, forming 1,3-biphosphoglyceric acid (BPG). Then 1,3-biphosphoglyceric acid will produce ATP when it turns into 3-phosphoglyceric acid. Here, 2 ATP molecules were produced. Then the 2-phosphoglyceric acid will turn into 2-phosphoglyceric acid, and finally, then turn into phosphoenolpyruvic acid. And finally, the phosphoenolpyruvic acid will turn into 2 molecules of pyruvic acid, where here another 2 molecules of ATP will be produced. Thus, this means the glycolysis reaction here produces 4 ATP molecules, and at the same time it uses 2 ATP molecules here. This makes the total ATP produced in the process is 2 ATP. Now we will learn on the fate of pyruvic acid. During the transitional step between glycolysis and the Krebs cycle, pyruvic acid is prepared for entrance into the cycle. If oxygen is scarce, or also known as anaerobic condition, then pyruvic acid is reduced via an anaerobic pathway by the addition of 2 hydrogen atoms to form lactic acid. This reaction regenerates the NAD+ that was used in the oxidations of glyceraldehyde triphosphate in glycolysis and thus allows glycolysis to continue. The series of events by which glucose is converted into lactate and back again to glucose is called the Cori cycle. The Cori cycle takes place in liver. When oxygen is plentiful or in aerobic condition, most cells convert pyruvic acid to acetyl coenzyme A. This molecule links glycolysis, which occurs in the cytosol, with the Krebs cycle, which occurs to the matrix of the mitochondria. The enzyme pyruvic dehydrogenase, which is located exclusively in the mitochondria matrix, converts pyruvic acid to a 2-carbon fragment called an acetyl group by removing a molecule of carbon dioxide. Each pyruvic acid loses 2 hydrogen atoms in the form of 1 hydride ion plus 1 hydrogen ion. The coenzyme NAD+ is reduced as it picks up the hydride ion from pyruvic acid. The hydrogen ion is released into the mitochondria matrix. The acetyl group attached to coenzyme A produces a molecule called acetyl coenzyme A.